Anis cannot see your face. Okay, so, right. Thank you everyone for coming today. Assalamualaikum warahmatullahi wabarakatuh. So today I'll be presenting, we will talk about this one paper that was published in 2022, personalized neoantigen vaccine and EOPV01 with chemotherapy and anti-PD-1 as first-line treatment for non-squamous, non-small cell lung cancer. Okay, so the reason I chose this paper because I wanted to know more about how people use peptide vaccine in treating solid tumor. Okay, and hopefully it will give us some ideas to proceed into this research area. Okay, so this paper it tells us it is actually a clinical trial. Okay, it uses the neoantigens, which are the tumor-specific proteins arising from somatic mutations that can serve as targets for immunotherapy. Okay, neoantigen is it's a protein or antigens that are produced from a mutated tumor, from a mutation in the tumor. So it could be served or identified by the immune system, especially the T cells, and perhaps our body can attack that tumor cells. Okay, so in this paper, they also combine the peptide vaccine treatment with the immune checkpoint inhibitors, which is the anti-PD-1. And also combination with chemotherapy. The chemotherapies that they use in this study are the standard first-line treatment for NSCLC. Okay, so the treatment with chemotherapy gives a good response among this patient, but however, after they get good response from this treatment, they detected a lot of neoantigens among these patients. Okay, so that's why they designed the vaccine based on these epitopes, the antigens, and they tried to do this research. Actually, there was another study. They designed the peptide based on this patient or based on this project, and they used for this research. I was thinking like I should have gone for that paper, but it was a bit too late. I chose this paper first. So maybe next time we should probably go back to that paper and see how they design the peptide vaccine in this cancer. So the objective of this study is to so it was the introduction before. Sorry, I did not have time to put in the PowerPoint. But the objective of this study is to evaluate the safety, feasibility, and immunogenicity of NeoPV01, a personalized peptide-based neoantigen vaccine in combination with pembrolizumab, the anti-PD-1, and chemotherapy, pemetrexed, and carboplatin as the first-line treatment for metastatic non-squamous NSCLC. So, for these patients, I don't remember how many patients, 60 plus or something like that, So they, before they designed the peptide vaccine, so they did the RNA sequencing and also exome sequencing to detect the mutations among these patients. So based on those mutations, so this is why it's called personalized peptide vaccine. So it's a bit costly, but if you notice nowadays in papers, in research, the world is going towards a personalized treatment, So this is what we were talking about, okay? Personalized peptide vaccine. Because, for example, even in the leukemia, one treatment doesn't fit all. So it has to be personalized because a lot of mutations. So the method that they use for this clinical trial is phase 1B, single-arm clinical trial. So they don't have a control. They have 38 patients, sorry, so it's not 62. 38 patients enrolled. It was described somewhere, okay? I don't remember where. Okay, it was enrolled at 4 clinical sites in the States, I think. And the vaccine development, as I said before, tumor DNA or RNA sequencing identified the neoantigens among these patients. And they designed the peptides up to 20 per patient. So it's not only one sequence of peptides. So it's a big vaccine, it's a big peptide. So 20 per patient. And then they did the intramuscular vaccination, started at week 12. So if we could go to the schedule here, which when you compare Hanis from the papers that you have seen before, this is much better schedule or clinical trial flow or schedule, it's much comprehensive. You could see here in the first phase of the clinical trial, they did the pembrolizumab Q3W. I don't remember what was that, but maybe a standard term for the chemotherapy treatment. Okay, so this is the pre-vaccine. So those patients here, they have not received any chemotherapy. So it's like a newly diagnosed patients for this type of cancer. Then in the pre-vaccine stage here, this is week before between 0 to 12 weeks in this clinical trial. So they receive the treatment, okay, and then they, after week 12 until week 24, then only they receive the peptide vaccine here. And then they monitored the patient 52 to week 52, so it's longer than a year, it's more than a year clinical trial until almost 2 years, 104 or exactly 2 years. Okay, so I think these dots here, the blue dots, the gray dots, and the orange dot indicate what types of sample collections that they took from the patients along the time here. Okay, so the study cohort, this is what they label their patients. Okay, initiated chemo, pembrolizumab, ITT set. I don't remember the full name of it. Last night I was trying to find it, but I found it. It's written in the paper here. But so when it says ITT, so this is what they are referring to. And then the vaccinated subset here, so it's a subset of ITT here, so they name it as a VAX, and then completed neo PV1 dosing per protocol cohort. Okay, and then you could see here the number of patients. They started with 38, and then there are some dropouts here, various reasons. Some developed the progression of the disease, so they had to withdraw from the clinical trial, 21 patients. And then the last analysis, the final number of patients, until the last final analysis, there were 16 numbers of patients. Okay, so these are some of the reasons of the dropouts. Okay, 7 not vaccinated, and then incomplete vaccination, and they did not include in the analysis. Okay, so the objectives that I mentioned earlier, okay, to evaluate the safety, visibility, and immunogenicity of the personalized peptide-based neoantigen vaccine. And also they wanted to see actually immunogenicity, vaccine-induced CD4 positive and CD8 T cell responses, okay. And they also wanted to check the epitope spreading, which is something interesting and new for me. Okay, to that the spread of the epitopes after we will look into the result, but after the vaccination or even before the vaccination, they measure the location of CD4 positive and CD8 positive cells, and then they predict the outcomes. The location, how many CD4 positive cells were there within the tumor or outside the tumor, can indicate or can predict whether the outcome will be good or not for the patient. So we will go through the result here. Okay, so these are the general characteristic of the patients here, so I will not go through this. Okay, we will go through this one, the correlation with clinical response or observed pre-treatment in the TME. Tumor, TME here stands for tumor microenvironment. okay, including T cell infiltration, major MHC class 2 expression, and T cell receptor diversity. Okay, so this is what I mentioned just now. First, we observe the infiltration of both CD4 positive and CD8 positive T cells into the TME at the pre-treatment time point. Pre-treatment, you know, it's not even vaccinated. Correlates with clinical response, suggesting the immune state of the tumor before the patient undergoes treatment can predict response to the treatment. Okay, and then further analysis of the specific localization of CD4 positive, CD8 positive T cells reveal the infiltration of CD4 positive T cells into areas with tumor cells correlate positively with response. CD4, CD4 positive T cells within the tumor cells, okay, and vice versa, CD8 positive T cells outside the tumor areas correlate with positive response among this patient. Okay, so I will not go through this. This is the progression-free survival that they predict to all these patients. I did not go through the detail for this one. Here, okay. So you could see here, so among this patient and 19 patients, okay, what they did, they took the samples within the tumor or outside the tumor, and they measured the CD8 positive T cells and CD4 positive T cells. So if it's outside the tumor, you see here CD4 T cells outside and within the tumor, the beta R correlation here is 0.68. So meaning that CD4 T cell positive, CD4 positive T cell within the tumor can predict a better outcome among this patient, if you get.
What's PFS now?
Progression-free survival.
Oh, okay.
Where did you see it? Because I also was trying.
On the top left side, this one.
Yes. So you see the blue dots here, no PFS, meaning that it's not progression-free survival. The green one is patients with progression-free survival. Free from disease. So they progress free from the disease. And you see the R here is 0.66, is much better than 0.36, although it's not perfect, 0.9, 0.8 or close to 1, right? This is correlation analysis. But
what's on the x-axis here? The y-axis, we know it's the cells.
Months.
On the x-axis, like
how long they progress free.
Oh, okay.
Yes. Yeah.
On the left side, the y-axis is cells per millimeter square.
Okay. Tumor cells per millimeter square. Okay. Okay. Right. And also, they took the example or tumor biopsy from patient 2L3, no PFS, meaning that it has a progression of the disease, and then the patient with PFS 9, which 2L5, which has a good outcome. Okay, and then you could see the CD4 positive cells here. I can't really see the green color here because to be honest, I don't understand this. Where is the within the tumor and where is not outside the tumor, because I'm not from solid tumor background, so I can't really tell here. Okay, but you could see here the difference in the colors. Here has a lot of CD3 and also the green color CD4 in comparison to the no PFS 9 here. Can you see? I assume this is within the tumor environment. Okay, right?
Why did they also measure CD3?
I don't know the function of CD3, to be honest. Do you have any idea of CD3?
It's like control, right?
No, I don't think it's a control. I think it relates to the CD4 thingy. So if you have more CD4 positive cells there, probably there's another marker that shows the ability of the immune response or relate to good immune response there. To be honest, I do not know the function of CD3. Okay, got it?
Yes.
Can we move?
Yes.
Okay, and then this is what is this? Wait, figure 3D. Okay. So figure 3D is the HLA-DR. So HLA-DR is the MHC class 2. So it will bind to CD4 positive T cell. So this could relate, CD3, the question that you asked, could relate to the enrichment of the immune system related to CD4 positive T cells. Okay, so no, they compared with no PFS with good PFS here. You could see it's obviously different, totally different markers here, So the red color is HLA-DR. So HLA-DR, they stain immunofluorescent on the class 2 MHC, meaning that it will present to the CD4 positive T cell. There are a lot of red color here, and also the PAN-CK. I don't know. I did not check for that. Sorry. The P is for the nucleus. CD11C, CD14. It's an indicator in monocyte derived cells. So monocytic. Okay, cells. So you could see the one with the longer PFS has better CD4 positive or immune response. Okay, got it, huh? This is pre-treatment, This is not without the vaccination first. This one I do not know. What is Shannon's trophy? Actually, I did not have time to check for that. But you could see here week 2 to 12, because they started the vaccination at week 12, right? But for the no PFS, you could see the count of what? The count of what it did. No, the count of cells, maybe the count of whatever. This one I think related to the CD4 positive T cell, because this figure is related to the CD4 markers, the markers of the immune response. But they did not show increment or just down regulation of these markers. Okay, but for the green color, those who have better survival, they whether like stagnant or they get better. Okay, most of them. There's only one case here. Understand? Okay, maybe we can check. This is figure 3F.
Legend down there.
Figure F here, analysis of channel points in tumor biopsy maturation. I don't know. Maybe they did not measure what they measure actually. Okay, but I think it's related to CD4 positive T cells. Okay, so now this figure is the conclusion from this figure. So we can, based on our discussion just now, we can say that a highly inflamed tumor microenvironment with high T in cell infiltration, MHC2 expression, because HLA-DR, is linked to improve clinical outcomes in this type of tumor. Okay, and also interestingly, CD4 positive T cells play a key role in giving a positive outcomes.
Inside the tumor.
Inside the tumor, okay. Especially tumors with strong MHC class 2 expression. So when you relate it, okay, right? Okay, now we move to the figure 4 here. Okay, so this figure is actually the measurement of T cell response after they vaccinated the NeoPV01 peptide vaccine. Okay, in combination with the chemotherapy anti-PD1 and anti-PD1. Okay. Actually, I was having quite a hard time to understand this figure. Okay, because they put 20 peptides for each patient, and this indicate the immunized peptide. So meaning that the peptides that should induce immunogenicity, in this patient. Okay, right. So this vaccine percentage of vaccine peptides, I also do not know how they measure this, to be honest, but from this figure, yes, the ChatGPT say 55% of vaccinated peptides elicited an immune response. So I don't really understand because this is the percentage of the vaccine peptide, but somehow it increases here. And then when you relate to figure B here, okay, here. So from this peptide, 13 patient, from this 13 patient, most of them, all of them, they generate the T cells response, the epitopes, right? And most, in comparison between CD4 positive and CD8 positive T cells, the highest one is the epitopes generate CD4 positive T cells in comparison to the CD8 positive, meaning that vaccines, if we relate to the figure 1, those tumor that has CD4 positive T cells within the tumor microenvironment, and this vaccine, when they injected the vaccine, meaning that the vaccine can enhance the CD4 positive T cells in tumor microenvironment. Okay, maybe I'm mistaken, but that's what I understood.
Yeah.
But makes sense, right?
But
Okay.
I don't understand percent vaccine peptides.
Yeah, this one I don't understand.
Yeah.
I don't
So these
understand.
peptides like exist inside
Yes.
the person.
I would understand if they put the percentage of T cell CD4 or CD8 positive T cell percentage here.
Yeah.
But I just don't understand here why percentage vaccine peptides, how do they measure?
Maybe they exist already inside the human from before, but is that possible?
No?
No, right? Vaccine peptide.
Maybe the pre-vaccine, they measure when they put it at that time, and then post-vaccine is after that. They put it.
Not what they put. That's what I don't understand. I understand the pre-vaccine and post-vaccine here. But what do they measure here? Percentage vaccine peptides.
In this thing, they said the peptide that elicited interferon gamma response.
Yeah, I understood that sentence correctly. So that's why I said if the y-axis here, if they put interferon gamma response, yes, definitely I agree. But here they put percentage vaccine peptides. So meaning that each individual, they receive 20 peptides.
So maybe
How do they measure the post-vaccine of percentage
Why is it
vaccine
going up?
peptides? How do they measure? No, you have to understand the unit here. Understand?
Yes.
Yeah.
And there's no 100% vaccine peptides either. It's going to 80 till.
What do they measure here? I'm just confused with the unit here. If they put the percentage of interferon gamma increment or something like that, CD4 or CD8 positive cells, I would understand. It makes sense. But here I don't understand. Okay? Right? Okay, so now can we move to the C here? So now they want to okay, now they took the patient sample from 2L7 and 2L3, and then this IM13 specific peptide. So IM13, IM0T is the number of peptide. Remember, one patient received 20 peptides, right? So with patient 2L7 here, they have this specific peptide. Patient 2L3 here, they receive the same personalized peptide for him, okay? So what they did here, they wanted to see if the personalized neoantigen peptide vaccine would further enhance the T cells response. Okay, so they took the PBMCs from the patients here, okay, and they challenged it with peptide in increasing concentration, okay, in the manner of increasing dose concentration, So you could see here, to L7 it has 2, okay, sorry, it has 2. This is to L3, so they challenge it with 2 peptides. So if those peptides is specific and could induce immune response, it would increase in what? Anis,
Sorry.
if that peptide is very specific to that patient and effective, and you challenge them with their samples with PBMCs, so it should increase in what? Interferon gamma, which they measure based on the
PBMC.
spot forming cells here. So spot forming cell is like the method, the ELISA or ELISA spot. ELI spot, sorry, the ELI spot. So it's you count the spot. You know, there's some reaction, chemical reaction.
That says, count the cell that says secrete the interferon gamma.
So you see it inside the cell or outside?
The cell itself.
Inside, right?
So it's in a plate, okay, and then you count the spot. So meaning that if more spots, meaning that you will get, it's a good, meaning that higher interferon gamma spots. You know,
Do you count the spot manually?
I do not know actually.
4,800.
I think it's a plate reader. You use it's a plate reader. Okay, understand now?
The mutant thing that they say here, the mutation.
Yes, the mutated
Peptide.
neoantigen, because it's a neoantigen, so mutant peptide.
Because wild type, of course,
Oh,
it
wild
doesn't
type.
take
So
the
wild
interferon
type-
gamma.
So wild type is the sequence of peptide that doesn't match anything. It's like SI mismatch.
Oh, okay.
So for your control also, if you want to do the peptide validation, you have to have a wild type peptide. It doesn't match anything in your in a human body. Must remember that.
Wild
Understand?
type- Wild type apa, doesn't match in human body or it
no, sorry.
native to human body,
Huh?
so that it doesn't trigger.
Yeah.
Sorry, say again.
I mean the wild type mean that the peptide is native to the human body, so it doesn't trigger response.
Yes,
Yeah.
that's better. That's more accurate. I remember Dr. Ng. I don't remember what was that actually. He mentioned about this during journal club. Journal
he
club
asked-
or
He asked like journal club.
who's
during
present?
his
Oh yes,
presentation.
So he asked you what's the problem with the study design here, because they did not use, they used the placebo control.
Mm.
Okay, right?
Yes.
So they did not use the wild type. I do not know how. Maybe they did not define the placebo. Maybe they have the wild type. I do not know. Okay. They just put the placebo. So you could see here in the manner of increasing concentration here, when you have the wild type, which the, say it again, Ameh?
Native.
It is native.
Yeah, native to the human.
So meaning that even if you increase the concentration, it doesn't give extra response here. But if you challenge these PBMCs with increasing concentration of the mutant peptide, it increases the interferon gamma response here. So it's very specific,
Mm.
right?
Yeah.
Understand? Okay, yeah? Got it, huh?
Yes.
Okay, now I just read from the analysis here. It says here 85% of vaccine-induced T cell responses remain detectable at 52 weeks, 29 weeks post-vaccination. Okay, some response appeared later, delayed activation, suggesting a prolonged immune stimulation. I don't understand here, to be honest. To be honest, I don't.
For that curve that you just showed us, doctor, why are there only 4 patients that they selected? What about the others?
Two patients. Sometimes in clinical trial you cannot show everything. It's very, you have very limited number of samples. You know, you withdraw the blood from the patients, but you only have for one set of experiment, and you don't have for RNA-seq data or whatever subsequent experiments, so you cannot.
Okay.
Doctor, I have a question. Like, for these 2 patients, right, doctor, why they choose only like 184 peptide only? Because they put like 20 peptide, right?
I do not know, to be honest. Maybe
20
they
peptide for
one patient.
One patient.
Maybe they have extra data in supplementary.
Okay.
Usually that's also answers you to your question. You know, sometimes you cannot put everything in the main figure you put in the supplementary. So you put those best figures, best result to represent the whole outcomes or the whole result from your study.
Regarding the figure A, they actually use 204 different peptides for that.
200 different peptide.
Yeah, for
This one.
yeah. So actually, they use 204 different peptide in 13 patients. And then they take the blood sample and then they see the same thing, the interferon gamma secretion. So that percent is percent from 204.
Oh, so 50. So 13
So-
patient, 13 points here.
30 patients.
13 patient.
So they did.
Also
of
they
30
count the
patient, and then the epitope generating T cell 50 from the 30 patient 55%.
Okay.
Yes.
Understand.
Yeah.
Again,
Yeah, say it again.
Because previously we know the 20 peptide kan. But then for this like ni they use 204 peptide. They want to see whether this peptide pre and post increase or not the response.
They did this ex vivo or in patient? Because I thought this is ex vivo, so they draw the blood from the patient.
Yeah.
They challenge.
Patient. Draw blood from patient.
And they tested with the 204, right?
Yeah.
so the challenge
In ex vivo,
in SVO.
not in patient. The
Yeah.
patient received the same 20 peptides, you know. So they withdraw the blood before the vaccination, and they did add another 204, right, Amir?
Yeah.
And then after the vaccination,
And what about post-vaccine then? Post-vaccine, they took the blood again.
yeah, they took the blood for a couple of times, right?
Yeah, in the other graph, percentage of vaccine peptide, serial PBMC.
So they take the serial PBMC pre and post vaccine.
Oh,
Okay.
So basically apa? Before vaccination, out of 204 peptide, very few that trigger immune response.
and then
After
post-vaccine.
vaccination, they again challenge the peptide 204 and then it increase.
Here.
Yeah.
Yeah, this makes more sense.
After 8 weeks.
So this table actually indicated the post-vaccine measurement, not the pre. So you could see here, you know, increase. So this is what they are measuring in table B here. Okay,
Yes, understand.
got it now. But this one is still like confusing, you know, percentage vaccine
Right.
peptide. Should be interferon gamma.
Yeah.
Kan?
But they just want to show like out of 204 how many percent of the peptide able to
Do you think this is 204? The dots here, it's just overlapping.
I think these are 13
Yeah.
13 patients.
1, 2, 3, 13.
13 patient.
Yeah,
Ah.
13
Before and
points.
after.
Yeah.
Okay. Okay, makes sense.
And then from that percent vaccine peptides are the 204.
Yes. Agree. Okay, can we move here?
Yes.
So here I read it again. I will read it again. 85% of the vaccine-induced T cell responses remain detectable at 52 weeks. So meaning that they took the blood samples and they measure. So what they saw is 85.3% vaccine-induced T cell responses even after 1 year, almost 1 year measurement. But I just don't understand how this actually in some of the, so they measured 2 times here, what they indicated in this table, week 20 and week 52. Okay, some of the peptides or did not induce any immune response in the first week 20, but then suddenly at week 52 they managed to get the response. So meaning that it's a long lasting immune response. that maybe comes later, you know, it's a bit
Take
delayed.
time.
Delayed immune
Yeah.
response. Okay, got it, huh?
Mm.
Okay, now
Cases.
Is that good or bad? Like delayed response? Is that
I think it's good for cancer resistant, you know. It's good to
like
prevent
prevent
recurrence of the cancer.
recurrence?
Recurrence.
Oh, okay.
Right.
But delayed response might not be good for example, if the
Treatment.
disease
With the
actually,
acute.
yeah,
Mm.
acute one, it's not
Mm.
good.
Mm.
But for like it's good.
Mm.
It's not good. It's just okay.
Mm.
I think in that one, if you see 2L11, that one shows like it has response, but then also delayed response. So that could be the best one, right?
Which one? 2L11. This one.
So this would be probably the best because it has response
Yes,
and
and
also
the
delayed.
delayed. Oh, now I understood this figure.
Both, yeah.
Now I understood this.
So till week 20 it has this much, but then week 52 it has more.
Mm.
So slowly increasing.
Yeah, makes sense.
But then 2L14, 2L16 are all later.
Yeah.
2L13,
2L14 and 2L16 actually.
yeah. And 2L13 could be short term.
It's not for the aggressive tumor, you know. It's not
Yeah.
good for the. Okay. So now they measure. Wait, is cytotoxic potential. Okay, now they measure whether the neo PV01, the peptide vaccine, has the potential of killing the tumors or not. Okay, so what they did is to they used the CD107A. Okay, it's a marker for T cell killing. So meaning that if there are signals of CD107A, meaning that cytotoxic is happening there. Okay, so I don't understand this. You know, they stimulate stimulation. I don't understand, to be honest. Maybe they mentioned it in the text. Okay, but what they did here is the ex vivo sample and then they stimulate with something. Okay, and this is the control, the MSO, and they did not see any increase of the interferon gamma. Okay, the y-axis is the interferon gamma. Okay, the CD8 positive T cells, and then CD107A is the indicator for cytotoxic capability potential. Okay, so when they induce or they treat the sample with specific epitopes here or antigen epitopes, neoantigen here, IM13 here, so you could see the increase of 56.2% of CD107A and also increase 16.2% of the interferon gamma here. So meaning that the neoantigen peptide vaccine here has the potential of killing the tumor cells.
But stimulation in computer, they like compare ex vivo and stimulation like actual and.
I don't know, maybe it's written. Did you write? Did you read in the? I don't know. Maybe it's written in the text, to be honest.
For
Find figure 4E or F, sorry. Did you find?
no, I didn't see anything about.
Go on. Maybe you can read in the method, you know,
Yeah.
you can check that in the method. Okay, but the key take-home message here, it's co-expression of the interferon gamma and CD107A display the cytotoxic activity of the neoantigen peptide vaccine. Got it?
Yes.
And also, I think maybe they did a individual. They challenge each patient sample with individual peptides. Maybe they put that in the supplementary, because they have this very comprehensive. How could they measure one by one and make a conclusion of this? So
It says
maybe they did.
5-day stimulation assay design.
Mm.
And ex vivo has.
Method.
Uh,
From here, it said neoantigen specific immune response were detected either in ex vivo or after 5 days stimulation. And for ex vivo assay, PBMC were rested overnight and supplemented with antibiotic. Then for neoantigen pre-exposure assay, PBMC were rested overnight and cultured with peptide or pool peptide for 5 days.
That's stimulated.
Okay.
Because ex vivo, if you see, there are no
Yes.
response.
Mm.
So stimulated is the one where we see the response.
Okay. So even the stimulated here without the specific peptide here,
Yeah.
they don't have the interferon gamma right here.
Yeah, 16%.
But when they challenge it with a specific peptide here, number 13 here, it has the interferon gamma response.
Individual peptide refer to
This.
I am 13.
IM30 the antigen, specific antigen.
pool peptide.
Pool peptide maybe the whole 20.
Oh, 200 for
They stimulated for 5 days and then
5 days.
they challenge with one specific peptide.
On day 3, half the well volume was replaced with a fresh media, and all the immunizing peptide were tested in both ex vivo as well as in the 5 day
see?
stimulation.
So meaning that they challenge with pool peptide first, then after that immunized peptide. So IM stands for the immunized peptide, specific peptide.
They said in total 92 vaccine peptide were tested on 12 patients.
12 patient sample.
Yeah.
So here the summary of the F here.
That's like 92.
So they found here
Ah.
92 immunized peptide tested here. 61% of from the 92 here has the cytotoxic potential, meaning that co-express interferon gamma and CD107A here, which is good to kill the tumor cells, right? It's
But
quite
CD4, 92% of it is CD4.
yeah,
90.
93% only.
0% CD8.
Yeah.
Both CD4, CD8, 7%.
Okay.
Yes.
CD4 are the helper T cells, right?
Helper T cell.
And they're mostly localized inside the cell, inside
TME.
the tumor.
Yeah.
Inside the tumor.
Inside the tumor.
So we have mostly those, and then they will respond more
Better.
better than CD8.
Yes.
My question, doctor, so if these cells can secrete the interferon gamma, why the number of CD8 like low? Because CD8 should be
Secreted by
interferon gamma. So
CD4 positive cell? No,
no.
they don't.
Maybe interleukin something, but no, not
Interferon
interferon gamma.
Maybe the 7%.
7% both CD4, CD8. That's in 4 out of 56.
Yeah, I mean they test like the peptide, right?
Yeah.
That 4 peptide actually.
So four peptides give both CD4, CD8.
Yeah, so interferon gamma could come from that 4 peptide.
Yeah, 4
The
peptides.
other 52 peptide just for CD4.
In the previous flow cytometer thing here, IM13. IM13 is the neoantigen, or is it the peptide like this one 2L8?
This patient's number.
Oh, this patient number.
Yes.
Okay.
IM stand for immunized. So they label the peptide immunized number 13, number 4. 14, you know. Okay.
Ah,
But that's a good point, Hanis. I don't see like just the 7% of the both CD4, CD8 positive would produce this a lot of you know, if you see here 16.2, or maybe this is just one representative that
Yeah, this just
produce a lot of higher interferon gamma. So, but here
I mean,
they
like,
measure the
CD4
interferon gamma.
CD4 can produce interferon gamma, but not in high level.
Like CD8 does.
Yeah, like CD8.
Maybe that's why it's 16.2 here. I don't know how high or how low interferon gamma response. This one, that's a good question, but need to read more, need to check more on that. Okay? Okay. Let's move to the next article here, the next figure here. Something that I learned very interesting last night. Vaccination can lead to epitope spread, where the additional tumor antigens beyond those included in the vaccine. It's written somewhere here in the text actually. It's very interesting. Yeah, here, okay, given the cytotoxic phenotype of many T cell responses, of many of the T cells responses against the neoantigen vaccine epitope, we speculated that tumor cell killing by T cells could reveal additional epitopes. So once you kill the tumor by T cells, and then whether it's a new, I don't know, new epitopes or the hidden epitopes, you know, boosting the overall immune response to the patient's tumor. So there could be additional epitopes. Okay, this concept known as epitope spread, okay, has been reported in a number of studies assessing the T cells responses to vaccine. Okay. Okay, additional circulating T cell responses to highly ranked neoepitopes that were not included in the vaccine, referred to as non-immunizing peptides, NIM, immune responses that were detected only at the post-vaccine time point, week 20, okay, were considered to be epitope spread responses.
Oh, that's why like previously when they put the peptide, some are not early on but later on.
Later,
Yeah.
so that could be
Non-immunizing.
okay. So across the 13 patients analyzed, 9 patients, 69% demonstrated the epitope spread. Interesting. Okay.
Basically, they had new epitopes coming out of the T cell
Yes.
after killing the T cell.
Yes, after T cells, after getting killed by the T cell,
Nice.
not coming from the T cell, coming from the tumor.
Tumor, yeah.
Yeah. Okay. So you count, if you count here, so there were 9 patients here that had the epitope spread, and 0 from none here. Okay, and then I think this one they challenge again the specific epitopes, non-immunizing epitopes here. Okay, NIM among these patients samples here, you could see that they could actually increase the spot forming cells or the early spot, or meaning that increase interferon gamma.
Only 2 of them have stars. there.
The star, it was mentioned somewhere in the text, I cannot recall it now.
Oh,
Okay, or it was mentioned.
usually star or asterisk is for significance, right?
No, not that type.
Oh,
Not that
okay.
type of star. There was star method, the green.
Oh, this one's green stars.
They study, no?
Green star dinner ex vivo responses.
This is the
Same figure.
Yeah. It's in the legend.
Ex vivo.
Ex vivo responses.
So the other are not ex vivo.
No.
What does that mean?
Yeah, I so don't understand. Okay, maybe check this later.
Yes.
But somehow the conclusion from here, the immunizing peptide is still able to trigger the immune response. Okay, and how many patients from this actually has the KRAS mutation?
Oh.
Okay, I cannot recall how many. One key point here, 5A and 5B, because they took these samples 24 weeks after the vaccination. So meaning that these responses were detected only after vaccination, indicating they were likely triggered by the vaccine. So the new epitope spread, actually triggered after the vaccination. So vaccine kill cytotoxic, it produces a different neoantigen.
Is it like a response for cancer to evade but backfired?
Maybe. Maybe. Maybe.
Just randomly.
I think right, you're right, we're probably right. I don't know, maybe there were already some studies about that, but we...
Epitope spread.
Yes, okay. Okay, 4 patients developed immune response to KRAS driver mutation. This one, 3 to G12C, 1 to 12GC. I don't understand here.
These are the mutations.
These are the patient with the mutation.
KRAS.
Okay. Okay, this is the one that has a better, because I checked carefully last night. Okay, we go to this name. Okay, so this is the one. So when this patient has a KRAS mutation, okay, 2L15 patient here, when it was immunized with the NIM101 mutant or challenged with the non-immunized peptide mutant and wild type, you compare here the diamond shape here and then the diamond shape here. Okay, here. So meaning that it shows a very specific tumor. This NIM101, it's very tumor specific.
For the KRAS mutation.
For the KRAS mutation. Yes.
That's interesting.
I do not know the mechanism, cannot really comprehend the mechanism behind it, because peptide is like very specific. Whether KRAS produces the similar
Similar
antigen. Pathway is another thing. This is because peptide vaccine is based on the antigen, specific antigen.
What are like most, like for example, like peptide vaccine for virus and stuff, they get it from the E1 and the region, kan? But for tumor, the peptide come from that secreted, tumor, but then is it like outside area or, because for virus we take the capside and stuff.
Yeah. In tumor cells, the neoantigen is specific to those cancers that has mutation. So when it has mutations, that's where they produce the specific antigen. Apart from neoantigen, and neoantigen comes from the mutation.
Yes.
There is another types of antigen in designing cancer vaccine. TAA, tumor associated antigen. I cannot recall the differences, to be honest. That's why I said I should have gone to that paper. Maybe Hanis, next time you go to that paper we discuss. It's a good thing that we are discussing actually, and overlapping the two cancer and peptide vaccine together. Okay, so I don't know. Another one is TAA. Another another one more. I don't remember. Cannot recall. It's in my head, but I cannot recall it. So it's based on the tumor. That's why they did the RNA-seq and the exome sequencing to find the mutation, and they designed based on that mutation.
But this peptide is not like specific. It's not belong to, like, for example, this peptide is belong to only secreted, but secreted by the cancer cell or the surface area of cell.
Oh, I don't know.
I don't know.
Oh, you mean what kind of structure of the cell?
Yeah.
Because you are imagining the virus structure,
Yeah.
so we know the location where.
Yeah.
But I don't think so here. I don't think so here because the problem with cancer cells, when the immune response trying to kill or to detect them, it's not like a simple virus. Like the virus, they present the antigen on the surface of the APC cells,
Yes.
but it's a bit more complicated,
For tumor.
more complicated than the virus. Okay, so that's why we really have to do the RNA-seq and exomes sequencing to identify that specific mutated region. And I do not know how they actually come up with the antigen. I don't know, to be honest. How do they know these antigens that are detected by the T cells? How they did the bioinformatics in the beginning of the study. They mentioned that. How do they select that? It's based on the previous paper, Otto paper or something. His name. Okay. Otto Warburg. I guess he's a German. Okay. So you see here, right?
Yes.
And also this again, they use the NIM01 mutated cells here, mutant cell. They challenged with the specific peptide here, and they see this CD107A increase in the CD107A and a little on the interferon gamma. Okay,
Yeah.
it's a bit complicated, huh? You guys know how to read flow cytometry? No, right? So this is the final figure, and then before this figure they have exclude all CD negative, CD3 negative cells, CD4 negative cells, and PD1 low cells. There must be a surface marker that they measure. Okay, so I did not go through to this. Sorry. Okay, can we skip this?
Yes.
I think they just extract the samples and they did the
Oh, these actually alleles. Okay.
alleles. Alleles, huh? Tetramer. Maybe.
It's usually allele or
Yeah, actually.
So this is the target.
Yeah.
Oh, so the IM13 is specific to the RB04.
Yes.
Progression-free survival, yes. Tetramer, I don't understand this. Okay. This is prediction, perhaps.
Oh, this is cool.
I don't understand this.
Amir last time he presented the other day in GC spatial metabolomics. So they were also looking at the metabolites where inside the tumor or outside the tumor. And this is similar but in immunology.
Okay.
This is cool.
Okay, so the take-home message from here, I think CD4 positive T cells plays an important role, key role in predicting. The more you have CD4 positive T cells within the tumor microenvironment, that would help you to get a better prognosis of the patients. And peptide-based vaccine, which is very specific, personalized peptide to this patient, could induce the immune response together with the cytotoxic ability of the cells to kill the tumor. And also the non-immunized peptides could reveal extra immune response and could relate if, for example, the patient has KRAS mutation, it would give a better result.
Specific response.
Yeah, specific response for that patient.
So you test a lot of peptide. You need to test for both early response and late response.
Late
So
response.
different.
This one,
It's
at least,
a big group.
I don't know how much money they spend here. That's a lot.
Cancer Cell article, so just
No.
the publishing would.
Publishing it would be around 20,000, 20,000.
Just the publishing is more than our whole research grant.
Doctor, sorry, I have something on my
Uh,
You know, just now that we talk about figure 4, why CD4 percentage is increased, I think because of the CD4 is helper T cell, right? Helper T cell, they can differentiate into Th1 and Th2. Th1 is more to pro-inflammatory, so it can secrete the interferon gamma. I'm just thinking that, you know, thing.
Okay,
So
that
I
makes
think-
sense.
Yeah.
That makes sense.
So the CD4 more change to Th1.
How do we differentiate between Th1 and Th2? Like, is there a marker?
Of course.
Yeah, also they have marker and also the cytokine. They will secrete different cytokines. Yeah.
There are markers for
Yeah,
Th1 and
there's-
Th2. Yeah. But maybe they spell it out here in the text
Paper,
and
yeah.
the supplementary. Really to have read the detail, but just then.
But
Trigger Th1 response.
NLSC, is it aggressive or non-aggressive?
I have no
Sorry,
idea.
what?
Sorry?
The
NSCLC.
non-small
Oh.
cell lung disease.
Lung carcinoma.
No,
I-
because of in tumor microenvironment in like more aggressive cancer, maybe it's hard for immune cell to penetrate inside because tumor microenvironment is very toxic and
Yeah.
only cell cancer can survive in it. So meaning that if we can penetrate into the tumor also can we can
They did not
sustain
mention about the the type of the cancer. You know, they emphasize more on the vaccine, the neoantigen.
Yeah.
Oh, CD3. That's what. CD3 is directly linked to CD4 T cell differentiation. So maybe that's how they tell that which is pro-inflammatory, which is anti-inflammatory. Using CD3, you know.
CD3 for what?
CD3 is in CD4 T cell. So maybe they could tell the pro-inflammatory and anti-inflammatory using CD3 gating.
Yeah, that's why
That's why they use
the
CD3. there.
CD3 here. So if you see here, you know, gated on CD3.
Yeah. So they took CD3, CD4 both.
Yeah,
That means
CD3 is like the bigger umbrella.
Yeah.
CD4 is smaller one, and then PD1 is specific one.
Because this one is solid tumor, so they can measure like infiltration of CD4. But if like blood cancer, for example, like
And bone marrow,
oh,
can mention bone marrow, but I don't think that you can get the tissue biopsy there. You can only withdraw the blood, bone marrow,
percentage.
do the analysis on the bone marrow, and you take the peripheral blood from, and then do analysis so you can differentiate between these two. Okay. I think this is very interesting paper for me to learn. Big paper. Quite need more time actually to read in depth about this research. However, it opens up different angle of the research actually.
Very thorough also the paper.
Huh?
The paper also very thorough.
Yeah, that's why when you ask me, you know, doctor, do you have any paper for me to present to journal club? Remember?
Yeah, so just also sometimes search cancer cells.
You go specific to the website,
Yeah.
right? You don't go Google.
I like their
I-
way of telling apa? They
Yes.
explain everything. That's why. Like,
Yes.
sometimes you go to they don't explain.
The reasoning, you know, in
Yeah.
like I told you sometimes, right, when we talk about writing paper, writing thesis, so the study was designed, and then when they came to the result section here, they wonder, you know, why is this happening? So that's the reason why they did the next step.
It flows very well.
It flows very well.
Right.
So
And also, I noticed the figures that they have are very non-generic figures. Like if you see the
Sorry.
flow that they had for the vaccine,
uh,
it was very non-generic because all of the others are like very basic sort of diagrams. But if you see the vaccine one that you were showing to Hanis,
mm-hmm.
it's this one.
Very specific, you mean?
It's very like
Detail.
unique, different sort of like a timeline that they put here. So in the top they had this line. All the others I've seen are not very clear. Like using different colors, using different
Yep.
subsections.
Yep. Agree?
Because usually people think Q1, Q2, you just have that same format. Like
No, not necessarily actually. You can actually, even if you submit to Q3, Q4 journal,